Okay, so we have done for DNA. So now we're going to look at this RNA. Okay, RNA stands for ribonucleic acid, RNA. So it's a polymer highlight, guys. Always remember we have DNA nucleotides as well as RNA nucleotide. So please always make sure that if RNA is a polymer of RNA nucleotide, don't say nucleotide only. Are you clear? So the word RNA must be there. If it's DNA, then you have to write DNA nucleotides. And RNA, usually they are single-stranded. Okay, single-stranded for RNA, usually. Okay. However, internal region of some RNA molecule may have complementary sequence that allow the strand to fold back and pair to form short double-stranded segment. Now, what does this mean? So, RNA, even though they are single-stranded, sometimes they may be able to fold, and then a small segment here, they may have complementary base pairing. Are you clear? For example, A and U. Okay, let me draw it out this way. So you may have A here, here have U, C, G. But they're still single-stranded, even though some parts of the molecule, they are having this complementary base pairing. So in terms of nucleotides, the sugar in RNA is the ribose, very similar to deoxyribose, but instead of having hydrogen atom, they have hydroxyl groups at 2' position. 2' means carbon number 2. Okay, at carbon number 2, OH, ribose. H, below the ring is deoxyribose. Okay. Then instead of thymine, so they have uracil. Uracil again is a pyrimidine, okay, that can form 2 hydrogen bonds with adenine. So basically, whatever happened to the thymine, uracil do the same. Are you clear? So U and A always pair together. So that's why in this diagram, you can see that U and A always pair together by forming 2 hydrogen bonds. So in this diagram, guys, in this diagram, so very clear cut, if I draw this structure out, you will know that it must be RNA polynucleotide. Why? 2 things visible here. The presence of the uracil. Okay, the presence of the uracil, as well as the carbon number 2 here. Can you see that? OH group. OH group presence at carbon number 2. So based on these 2 features, I can very, very confirm and say that this is the RNA molecule. Are you clear? Another thing you can say that, oh, single-stranded. Are you clear? Another thing we can say that is single-stranded. DNA must be double-stranded. But sometimes we also can denature our DNA. We take out 1 strand. So that one is not so conclusive enough. But if you look at this, uracil and also the OH group at carbon number 2, definitely RNA. Okay? So highlight this activity 10.6. Activity 10.6 actually is very, very crucial because so many years, I mean, this question keeps on repeating by CIE to compare and contrast between RNA molecules and DNA molecules. So first, in terms of the structure, so RNA is a single-stranded molecule and has a shorter chain of nucleotides compared to DNA. DNA much more longer. RNA actually much more shorter in terms of the structure. DNA, typically a double-stranded molecule with a long chain of nucleotides forming a double helical structure or double helix. Okay? Now, next. How about bases? What are the bases involved here? RNA, so adenine, guanine, cytosines, and uracil. But DNA consists only of adenine, guanine, cytosines, and thymine, no uracil. Okay? In terms of base pairing, now, both of them, yes, both of them also have base pairing. But base pairing in RNA will be A with U, G and C. In DNA, it will be A and T, G and C. Okay? In terms of sugar, RNA, the pentose sugar is ribose. In terms of DNA, deoxyribose. Okay, deoxyribose. In the ratio of purine to pyrimidine, RNA not necessary to have the proportionality. Because A no need to pair with U. C no need to pair with G. Are you clear? They are not compulsory. No obligation for them to pair up. Only a small segment pair up. So in this case, they do not need to be 1-to-1 ratio between A and T, C and G. Are you clear? But in DNA, because DNA, A must pair with T, C must pair with G. So means that A and T, the ratio must be 1-to-1. C and G must be 1-to-1. So in this case, it must be 1-to-1 in terms of purines to pyrimidines ratio. Therefore, only we can maintain this what we call the parallel property of the DNA. Okay. So in our syllabus, actually RNA, we have a lot kind of RNA nowadays. Okay, but for us, only 3 kinds of RNA we're going to learn. Okay, and all of them involved in the protein synthesis. So not so soon we're going to learn it until we come to the part of protein synthesis towards the end of this topic. Are you clear? Because the next subtopic we're going to talk about will be the DNA replication. Are you clear? Clear? So now, what kind of RNA we're going to learn here is, if you look at this, they say that one of the structural components of ribosome, now we know, ribosome, synthesized by nucleolus. So synthesized, the nucleus synthesized are RNA, which form the ribosome subunit together with the protein complexes. So answer for the first one, rRNA. Now next, it acts as a codon by carrying genetic code which is transcribed from DNA in a nucleus to the ribosome. Okay, they transcribe. So involved with the transcriptions. So this one must be messenger RNA or mRNA. So next, at the end of this RNA is a site to which an amino acid can attach to, and at the other ends are anticodons. So okay, 3 unpaired nitrogenous base. So this one little bit, you guys haven't learned about this before, but again, guys, you can note that this RNA is special because they carry or they transfer or transport amino acid. Okay, so that's why they are transfer RNA termed as tRNA. So these 3 structure, RNA, mRNA, and tRNA, all of them involved in protein synthesis, or more precisely, polypeptide synthesis, which we're going to learn at the 10.3. Okay, our notes 10.3. You will see this all of this structure. Okay, we're going to study a ribosome in more detail. We're going to study about how the formation of mRNA and how the tRNA actually carry or transfer the amino acid in the process of polypeptide synthesis. Are you clear? Okay. But in fact, if you go through and search about RNA, we have about 20 types of RNA, a lot of types in our body. Some we use for research, okay, short RNA, okay, so snRNA, SNP, so a lot. But for us, at this moment, we're going to study about these 3 types of RNA first. Okay, clear? Clear? So with this, I've done for the first part of the lesson. So let me off the recording.